Well, hi everyone. Well, it's great to be back. This is literally my first talk since 2019. I'm Nicola Bordin. I'm a research fellow at University College London, and today I will explain to you what is CATH and what it is. And how we're expanding it with AlphaFold data. So CATH is a protein structure classification resource that was created in the early 1990s. And over the past 25 years, we processed protein chains from the PDB and into constituent domains, and we classify them into homologous superfamilies. So this process consists in kind of gathering them into common evolutionary ancestors, and then we classify them into a hierarchy that consists of CATH, hence the name, which is class, architecture, topology, and homology. And this classification is made using expert manual curation, and then which is guided by automated algorithms. This leads to, this is kind of our structural core of the database, and then we expanded in our sister resource, which is Gene3D, and we encode this evolutionary signal that we find in profiles into HMMs, and we build these HMMs for all these CATH structures, and we query the sequence space in UniProt using these hidden macro models, and we inherit all these kind of annotations such as Go, EC terms, drag bank, and OMIM, and we assign this to predicted structural domains based on sequence, and we're able to expand this core of structures with 150 million domains from UniProt. So CATH has two types of releases. One is a continuous release that is kind of updated weekly, and we fetch protein structures from the protein data bank, and then we chop them and classify them into superfamilies. But and you can access the latest up-to-date annotations from CATH every week. But once every 2 years, we freeze this process, and we generate a whole lot of extra release data, such as models, alignments, functional families, functional annotations, and so on. And the latest release was released in 2019, is CATH 4.3. So what you have with CATH B, which is the rolling release every week, you have the very latest PDB annotations. We have some automatic and minor annotations. Sometimes you may find some minor changes on a weekly basis, but whenever you have the CATH Plus release, you have a lot of this additional stuff, which is extra curation. We have some additional analysis. All this data is then shown on web pages, and we create also and distribute some data sets of non-redundant structural domains at 20% sequence identity and 40% sequence identity. And we make them available both as PDB files and also as classification lists. All the code that we use, it's open source. It's available on GitHub. It's called CATH tools, and you can use our own tools that we use for aligning, superposing protein structures, or resolving sequence scans using HMM libraries, or there's a whole lot of documentation on our APIs too. So our latest release in 2018 was CATH 4.3. And that consisted of 158,000 PDB structures chopped into 500,000 individual domains classified in 5,600 superfamilies. So this is just a general idea of the size of the data that we accumulated over 25 years. But then on my birthday, literally on 2021, AlphaFold decided to drop on us literally 15 years worth of protein chains. So the initial release was for 365,000 structures for 21 model organisms. And there's been some subsequent releases for Swissprot and organisms causing neglected tropical diseases. And there is a rumor that they're going to release UniRef 90 models for the whole of all UniRef, basically. So that's almost 100 million chains. So with the current protocol that we have at the moment, we can't really deal with this kind of data because it's kind of built on, besides this kind of algorithms that have been built over a very long period of time, we also have a lot of manual curation on these things. So we had to come up with something completely different. So we came up with CATH Assign. So CATH Assign, it's a protocol that takes these chopped AlphaFold chains and uses assignments from hidden Markov models and new machine learning-based methods. And then in order to deal with varying degrees of homology, we use a cascading approach where we scan and we validate over larger and larger data sets. What's remarkable is that we went from algorithm design, benchmark, data processing, assignments, curation for around 15 years worth of structures in 8 months. And now that the infrastructure is in place, we're able to cope with further releases of AlphaFold2, hopefully. So the first step in this is our internal tool, which is called CATH Resolve Hits. So as a part of this process of generating Gene3D, since we scan all these UniProt sequences against our HMMs, we have CATH assignments that can be detected by HMM for both CATH and PFAM. And we basically feed all these hits into CATH Resolve Hits. And this tool finds what is the best set of non-overlapping domains over an actual chain. And using that, we're able to find the boundaries of all these individual domains. So while for hits that have hits on CATH, we can use the HMM hits. And for some of the PFAMs and NewFAM domains, NewFAMs are domains where we don't have a CATH domain, we don't have a CATH assignment or a PFAM assignment. So these are things that we never seen before. We need to come up with something that is more powerful than HMMs. And in this case, we teamed up with the Rust Lab group at the Technical University of Munich, where they helped us build a CATH superfamily predictor based on embeddings from protein language models. And this tool outperforms HMMs. And in the case for the largest superfamilies in CATH, which are basically 50% of CATH only falls within top 200 superfamilies, we're able to assign close to 90% confidence to these domains. But even though we have this tool, we're still talking about 800,000 domains at this point. And we are interested in bringing in only things that will belong to CATH and will belong to PDB. So we need to set up some pre-filtering criteria. We use several. First of all, we filter on PLDDT. Anything that is below 70 gets discarded. Also, domains that are very tiny. So if they have less than 3 secondary structure elements, also they get set aside. And then we look at residues that are not in secondary structure. If 65% of residues are not in a secondary structure as predicted by the SSP, we also throw them out. We look also at long stretches of unordered or low quality regions, or the packing in the core needs to be kind of conserved in this case. So by using this kind of filtering criteria, we're able to, this is the kind of the composition of our discarded domains. You can see that most of them, around 52%, kind of very much in line with what Mike Sternberg announced before, that 50% of domains in the AlphaFold dataset, it's of low quality. 50% of them, they get discarded because of PLDDT, and then there is another 23% that are very tiny, tiny domains. As you can see, this distribution on the left, it's for the whole data set. On the right, it's for the 21 model organisms. More or less, the distribution is similar across all of them. So if we combine this with the good model quality, we can see that there are 52% of domains that are deemed of good quality, and they pass all these different criteria for being brought into CATH. So, but we're still talking about 370,000 domains at this point. So in order to do all these scans, we teamed up also with the Steininger team at the Seoul National University that just came up with this amazing ultra-fast aligner called FoldSeq, and it's incredible. Basically, you can scan 500,000 protein structures against themselves in probably 2 hours, which is very remarkable. And over time, while the first initial releases, they weren't as accurate. Now it's comparable to very old, well, kind of old state-of-the-art tools, and it's comparable to Tiamaline in terms of precision at this point. So for our project, basically we benchmarked it on a test set of 4,000 curated gold data set of 3,000 domains that have shared boundaries and superfamilies both in CATH and in SCOP. And then we looked at doing an all versus all comparison if we, at what point we would get an actual correct assignment, knowing previously what was its homology status. So we, also we tailored these thresholds on the CATH class in this case. So we have different thresholds for domains that belong to CATH, mainly alpha, mainly beta, or mixed alpha beta. And by using that, we were able to kind of bring in all these domains. So the next slide is based on those 52% of good models. And we can see that we're able to bring in 92% of them into CATH. The vast majority, as expected, they come from very close homologs, so CATH hits that came from CATH HMMs. Now we have still an additional 8% that were CATH predictions made from this machine learning predictor and then validated by Fold2 or SAP. And then we still had another 10% that CATH wasn't really confident, but by scanning against all of the whole of CATH and domains that were previously brought in using those 73 plus 8, we were able to bring in an additional 10%. And this left us with 8% of structures that were unassigned. So this is a massive increase in structural coverage. As you can see here, especially for certain organisms, plants mostly, you have Rhiza sativa, maize, and Glycine max that they have almost a 4,000-fold expansion in structural coverage. So also for our internal kind of classification, it was very useful because within CATH superfamilies, we have there are enough diversity in sequence between paralogs that you can find also different functions among them. So within superfamilies, we create a subset called functional families, or in short, FAMFAMs, where we're able to segregate these two kind of sequences, these two kind of functions, by looking at the presence of specificity determining positions in the alignments. But while these are great for function prediction or kind of looking at pockets, variants, most of them, they don't have an actual structural representative, only 8% of them. But by using AlphaFold data, we're able to expand it from 8% to 30%. And this one is going to be pretty useful in the future. So I'm going to go back to that 8% that we weren't able to classify. And these are either things that probably are not in CATH or not in PDB or something completely new. So we looked into it, we clustered them, and then looked at the 618 clusters that had at least a human representative. We did extensive manual curation on it. And we found 26 new folds. Most of them are alpha. But then we also find some very peculiar ones, like that heart domain. We call it the heart domain. And then there's a very peculiar alpha-beta propeller over there. So all of this data is going to be made available. It's not going to be part of CATH, so it's not going to get merged with the current classification, but it's going to be made available as a separate download. We're currently making the release packages. It's going to be ready probably by Thursday. And we're going to make the data available through Zenodo organized by either by superfamily or by organism, as well, and putting them on the CATH FTP. All individual models are going to be available through the 3D Beacons network, and it's going to be also made available via other venues. And at the moment, we're currently processing SwissProt with neglected tropical diseases next. So with this, I would like to thank you for your attention. I would like to thank especially Jens Silito for his help in setting up the pipeline, Vamsi and the team at the Technical University of Munich for the machine learning predictor, and Vishali for her amazing work on the curation process, and Sudat for benchmarking our tools on PDB, and as well, Martin and Stephanie for helping benchmarking Fold2. Well, thank you.
Very nice work. Thank you very much.
Thank you.
I missed the detail. As I understand, the PLDDT score is per residue.
Yes.
So when you say that you throw away a domain, if the PLDDT score is less than 0.7, what is it for any residue, the average of all the residues?
The average, yes. We average PLDDT, yes. And also some of the, whenever we look at these long unordered regions or regions of low quality, we look at stretches where the PLDDT is lower than 70. Maybe it can be, overall, it can be higher than 70, but if it's a stretch is it has bad model quality, we're gonna just discard it. So we use it also at the residue level for those stretches.
If you were to throw away anything that has at least one residue below 0.7, you won't have much left.
Well, but we don't throw things that just have one residue. There needs to be at least 30% of the whole domain.
Well, thank you very much for your presentation. First of all, as someone who's been using CATH for the last like 6 or 7 years, a big shout out for everyone who And my question is about the novel domain slicing tool. As we all know, the fact that we have domains, more than 6 domains in CATH is very nice. And when we're talking about incorporating the whole ref 90 into CATH, I'm concerned about the high amount of work on proteins where we cannot like have a reliable HMM assignment. So have you benchmarked your new, I think it's protein bird that you said, and like your new domain assignment domain slicing tool on those orphan proteins, and what are the results that you're getting from it? Like is it working well on proteins that have no possible assignment for experiment glory though?
So one thing we noticed is that for all these predictions where we have new folds, these things, they are hits where CATH, which is basically based on a PLM method, very similar to protein bird. It's literally based, the model is called PROT-T5, which is based on PROTBIRD Tucker, based on PROTBIRD, sorry. And so more or less it's the same thing. So at this point, it's the confidence that we take, it's for the superfamily prediction. So maybe the tool can be a little bit confused between superfamilies, but in this case, it's confused also at the architecture level or at the fold level. So in that case, even these machine learning tools, they're not really that useful at that point. So we're talking something that it's even using structural aligners where we're literally trying to scan that thing against everything. It doesn't match. So these are things that either we haven't seen before, and probably they're just sitting on on the PDB, and they need to be structured. They need to be kind of processed. But we kind of erase that because we did scan against the PDB. So these are actually new, I've never seen before.
Okay, thanks for clarifying.
Sorry.
Very nice presentation.
Thank you.
And my question is similar to the previous question about novel structures we might see an increase through AlphaFold models, novel domains, and how to classify them. So as I understand, mostly it is still a manual process to create this or curate these new families and classify them. Are you guys thinking about, maybe making some new platforms to crowdsource more people to look at these structures and classify them? Or you still think, it's not a bottleneck with this deluge of data?
Well, that's a very good point. We don't know. I mean, because at this point, we would expect, if you notice that all of the good models quality, we're kind of focusing on that 8%, but 92% of them were brought in. So whenever we're going to get all the next releases of AlphaFold and we scan them against even things that we expanded now with AlphaFold, we're kind of stretching the structured base even more. So probably we're going to kind of be able to find remote homologs just because we have the AlphaFold models that we classified already. So we can kind of transfer the homology through that. So we'll see how hard it's going to be. Pretty sure it's going to be hard, but it's a very good point. Yeah, crowdsourcing, it's a good idea. Yeah.
There's a virtual question as well. So this question is from Michael Sternberg. Why did you not use a structure-based approach to define domains could the unusual folds be errors?
Well, we did use the same approach as we usually do for doing this kind of these assignments. I mean, the data set, if we had to do structural alignments just to detect domains or like our usual tools, it would have been too slow, absolutely too slow. So the way we've done it, we made these predictions and then we validate them by scanning them using a structural aligner. But we can't really do kind of the assignment beforehand by scanning against everything.